Okay, I think we can start. Welcome everyone in 2024. I hope it will be a very good RNA year for everyone. And we start today with Joe Yesselman, and then we can go back to our regular schedule. So Joe did his PhD at the University of Michigan, and this was 2013. And then he joined the lab of Rijudas at Stanford. So this is where we met, and we worked a little bit together when I was with Riju, and then Joe worked on a RNA-make tool. So this is a tool that can be used to design RNA sequences that they will fold into given shapes. And in 2019, he started his own group at the University of Nebraska, Lincoln. And today he will talk about high throughput determination of RNA's tertiary structures dynamics by quantitative DMS chemical probing. So I see that his pursuit is consistent about the molecular dynamics of RNA. So the floor is yours, and I'm looking forward to your talk, Joe. It's good to see you again.
Yeah, thank you,
All
Magnus.
these years,
I
yeah.
really appreciate that intro. Let me share my screen. Okay. And hopefully in the 5 minutes since we tested it still works. Good. That's a good start. Okay. So yeah, as Magnus said, I was a postdoc with Riju Das. I did lots of RNA design work. But in my own lab, I still do design, but I've also become very interested in understanding tertiary contacts, which of course I'll give you a bit of background on. And so this is a new method that I've developed, which I'm very close to the preprint. Hopefully it'll come up in a week or so, using our old friend TMS. So let me give you some background. I'm sure most people are very familiar with RNA, but just for people who are not, so structured RNAs are extremely critical to the cell. Lots of the largest machines inside the cell are composed of RNA, usually with some proteins. So we have the ribosome, which translates mRNA into proteins. We have telomerase, which caps the end of our chromosomes. And then we have the spliceosome, which helps to mature mRNA by removing its introns. And all three of these are, you can see, are these large machines composed of RNA, and all of them have to be in intricate three-dimensional structures for them to perform their function. So thus, as many people here are interested in, we must understand how RNA can do this. How can RNA go from a single strand to fold into these extraordinarily beautiful structures to perform their functions? So RNA is very interesting in the sense that it has these rather discrete steps of folding. So RNA starts as a single strand composed of A, G, U, and C. And then it undergoes a rapid collapse to form base pairs. And we call this a view, where we're just looking at the Watson and Crick pairs secondary structure. Then secondary structure, excuse me, forms other long-range interactions that hold the RNA into a well-defined 3D structure. Now, of course, not all RNAs have tertiary contacts, but many of the functional RNAs that have catalytic activity or bind ligands, do have these long-range interactions. So for us to understand how RNA performs its function, we must understand how RNA can get to this 3D structure, and more specifically, how RNA forms these long-range contacts for it to be stapled into these beautiful 3D structures. So we think at this point that we can predict secondary structure, which of course, if you have been following the recent work by Riju and others, we can't 100% do this, but generally we have some pretty great rules developed by Turner and colleagues, which of course hopefully will be improved further. But really, we really still struggle to predict 3D structure. Now, again, we've made great strides. I was really impressed with the recent CASP results. but we still have a long way to go. I think we're still about 5 to 10 years out from this. And to me, there's two things that really are, I think, holding us back. One is RNA is extremely flexible. And so we need to really better understand how RNA can flex and fold on top of itself. There are areas of extreme flexibility compared to proteins. And then the other thing is as I said earlier, there are these there are sparse tertiary interactions. So these interactions, maybe even a big RNA the size of 500 nucleotides might only have 2 of these things. And these tertiary contacts are hard to predict and hard to know if they will form RNAs. And that's a very difficult problem because you need to know if 2 things come together, then you need to know the stability of it. So, and we've seen in this CASP and previous RNA puzzles that if you nail the tertiary contact, you do way better on prediction than if you don't. So understanding if a tertiary contact will form and its strength is critical to our ability to find the lowest energy structure of large RNAs. So it's not just this P4, P6 RNA I showed. Tons of RNAs from small to very, very large. Of course, I'm not showing the ribosome and others have tertiary contacts, but you can see RNAs throughout many different functions. So we have SRP, signal recognition particle, different ribozymes, riboswitches, RNase-P, group 2 introns. They all have these tertiary contacts. And you can see even really large things. I think I'm missing a highlight here for RNase-P. Only have like 1 to 2 tertiary contacts. And that greatly restricts the potential conformational space that can be in. So I'm always really surprised by things like SRP and some riboswitches. There's a purine riboswitch right here, where it's really just one staple that's really helping it stay together. Now, again, not all these tertiary contacts, they are often forming, they're all often performing the same role, but their stability varies greatly. Oops, can I go to the next slide? Yes, I can. Okay, so we have things like the A minor interaction. This is where A or multiple A's kind of flip out and form a triplet. So the A's either form, usually it's not a triplet. Usually it's just it's forming hydrogen bonds to the backbone or the minor group. So these are relatively weak. They're unlikely to hold a RNA into a complex 3-dimensional structure, but they might add additional stability. Then we have the tetraloop receptor, which is my favorite, which is a relatively strong interaction, probably on the order to 3 kcal per mole-ish. So these can, usually with help from another tertiary contact, restrain a large RNA. And then we have things like the Kissen loop, which is basically just a series of Watson and Crick base pairs between 2 loops. So these things are extremely strong. They have a delta G of 12 kcal per mole. So they can really hold things together. And there's lots of beautiful nanostructures by Ebbe Anderson and Cody Gary that utilize these to staple really large RNAs together, usually more than one because they're so big. So we need to both understand if for a tertiary contact to exist in a given RNA, we need to know if it can come together. But if it can come together, how strong will it be to hold the RNA together? Maybe it can come together, but it can't actually stabilize the RNA. So that's why thermodynamics is super important. And that's what the rest of this talk will be about. How can we measure the stability of a given tertiary contact? So, I mean, this has been a long-standing question. People have been interested for this for a very long time, back to the late 80s, early 90s. There have been 2 common methods for people to do this. And of course, I know I'm excluding some. There are definitely others. So one is single molecule FRET. Basically, you have your tertiary contact, you put a little tether on it, and then you allow it to dock together, and you will see quenching between the 2 dyes. Works very well. It's great. Then you have gel electrophoresis, where you can do native gels and you can observe, you can do it with and without magnesium, or the easier way to do it is just do 2 different mutations. So one can form, one cannot. So here's a wild type. This is from a paper I published, and here is a UUCG mutation. This tertiary contact cannot form now. And what you can see is a change in running. So the UUCG is less stable, so it's like flopping around like this. So thus it goes through slower. So you thus can see this, and basically how big this gap is directly related to their stability. Okay, so these again are great. They work fine, but what if you want to do more than one RNA at a time? I mean, yes, I'm sure some graduate students that are dedicated can do hundreds of these over their entire PhD, but there are, of course, hundreds, thousands of RNAs we're interested in, and more are being discovered constantly. So this is not going to work out, and we're going to save graduate students some time. So more recently, Dan Hirschlag and Will Greenleaf have developed a high-throughput way of measuring tertiary contacts. So this is a beautiful system developed by Luke Yeager and colleagues, where he has basically 2 tertiary contacts. So the trick was you could put one of these strands, this is called the tecto-RNA, you put one of these on what's a sequencing chip, you basically transcribe off of it, and then you flow in the other half, and the other half has a dye on it, and you can just measure association based on increase in fluorescence at a given spot. And you know what each spot is because you sequenced right before. So this is a really, really beautiful method. It is definitely a huge step forward, but unfortunately, it is really hard to do. You need this hacked Illumina sequencer, which I think 3 exist in the world now. And of course can't be supported because you're not supposed to do it, and you're relatively limited to what type of constructs you can do. This worked out really beautifully, but what happens if I want to look at a kissing loop or something like that? Well, I don't know if this scheme will work. So my goal in this project was come up with a easier way of doing this that could give us more information, and someone like me who is not very good at experiments could be able to do. So let me introduce that idea. So I'm all about DMS chemical mapping. Chemical mapping is an extremely easy technique. Even I can do it. I think basically anyone can do this experiment. It's extremely easy. So basically how it works is we expose RNA to DMS. It methylates A's and C's when they are solvent accessible, or at least they have to be solvent accessible. There's probably other factors. And now, if people have seen, Ribonanza had it. There is a new method developed by Musto and colleagues to now do G's and U's too. So it's extremely powerful. I won't be talking about that, just A's and C's. And then you can use a very special reverse transcriptase. In this study, we use TGERT, but you can also use Marathon, where these methylation sites shown in red, they will be converted to mutations. Given basically the reverse transcriptase freaks out, is like, I don't know what to do. Let me just throw in some random nucleotide here. And so then you now convert your problem to a sequencing problem. And sequencing problems is a solved problem because sequencing is very cheap. So this new method is called qDMS-MaPseq, developed by Sylvia Ruskin, who's now at Harvard. It is extremely powerful, and it allows you to multiplex hundreds, thousands, millions of RNAs. There's basically no limit, just how much sequencing are you willing to do. So I've always been very interested in this. What can you extract out of this technique? Most people, I think, know about it from a secondary structure. It's definitely worked great for figuring out secondary structure, but maybe fewer people know it actually works great for almost all tertiary contacts that I know about. So the thing is that because it's not just reading out base pairing, it's reading out some type of solvent accessibility. If there's a change in solvent accessibility, it can also work. Basically, you're just looking for a change, right? So here, we'll be talking about the tetraloop receptor, TTR. I'm not consistent. Sometimes I call it TL-TLR. It has 4 A's that are really nice probes. And there's a large body of research showing that when the tetraloop receptor is formed, DMS cannot get into these four into this complex. Basically, the tertiary contact blocks DMS from getting in, so you get extremely low DMS. Looks basically like a paired Watson-Crick paired residue. On the other hand, if the tetraloop receptor does not form, either by mutation or by different conditions, you get really nice reactivity. And again, this is not just true for the tetraloop receptor. It's true for the kissing loop. It's true for some A minor interactions. It is very useful to do this. And what's great about it is it's completely generalizable. And we're just sticking into an existing protocol that we know works. So here's the system I started with. So this is a RNA nanostructure that I developed in my postdoc. We call it a mini TTR because it is a very small, it is the smallest solid structure that we have that has the tetraloop receptor. It's about half the size of P4P6. So it has this topology, these three motifs, structural motifs. It has a kink turn, basically a IRES, it is part of the IRES structure from HCV, and this 3 and 3 loop. Okay, and here's the 3D structure over here. So it has this really nice, simple, like cloth pin-like structure. It's perfect for what I'm interested in because with the structure, we can make changes, and we have the known 3D structure at least as a starting point. This was crystallized by Jeff Keefe for our paper on this. Okay, so what does the DMS look like? So the coloring is blue is low reactivity, gray is no reactivity, i.e. G and U in this case. And then red, going up to red is high reactivity. So you can see we're looking at our A's down here. You can see there's basically no reactivity. There's a little bit of reactivity in here. That is likely just the conditions. This was very old data. I should probably remake at this point. Then you can see other places, so this A here, this C here, they appear to be very solid accessible. So this is basically our starting point. Okay, so the other really cool thing, the other piece of this puzzle is there is a huge body of research showing that at physiological conditions, if you change the magnesium concentration, you can affect binding of formation of the tertiary contact. So here is a very old chemical mapping run I did using the stop method. Not using this anymore because it's kind of a pain in the butt. But if you can see the tetraloop receptor, if you start at zero reactivity, zero magnesium, you have high reactivity. Dark is more reactivity, and light, it goes to light, no reactivity. So you see the case for the tetraloop, you see the same for the receptor. So my thought was, well, okay, we have this we have DMS. We know we can titrate magnesium. Seems like a very solid bet. Now that we have these multiplexing techniques, we can now look at hundreds of RNAs at the same time. So that was my thoughts. So starting my lab, this was some of the first data I collected from when I started, is I did basically replicated that RT stop method, so the older chemical mapping method, and did it with DMS-MaPseq, this newer mutational approach. So I look at 40 millimolar magnesium just to make sure. I see, so here is where the tetraloop is. Here is where the receptor is. So you see for these three A's and this A over here, they're very low reactivity. So I'm like, great, we got it. But let me just make sure. Let me change it to zero magnesium, right? So I showed you previously with zero magnesium, it does not form. It does not form. That's the beauty of this. But when I did this, it looked basically identical. And at least in the tetraloop receptor area, so in these red highlighted areas. And I was like, what? And I must have done this experiment like 3 times. I put things through a extra column. I was looking up columns for removed magnesium. I'm like, wow, something's really wrong. So I started to think of other things. I'm like, well, what else can we do? well, we could change the buffer. Maybe this is some kind of weird buffer thing. So the common buffer we use here is sodium cacalate, is from the original papers on this technique. I tried HEPEs, I tried tris. Basically looked the same. I mean, yeah, tris was a little bit higher, but at this point, I'm like, wow. I mean, I really don't get what I'm doing wrong here. So I do this like really large. I'm just trying like every condition. So basically, what we're seeing here is we're seeing the buffer and the concentration of the buffer. So the standard concentration for these experiments is 300 millimolar buffer. Now, this came from the original paper that thought up this kind of mutational profiling approach from Kevin Weeks, and he showed that he needed this high buffer because there was more DMS and that it would basically quench itself if you had lower buffer. So I was like, well, first, is that true? And you can see, so each one is, what you're seeing here is a correlation between the reactivity, so reactivity at each position between the two conditions. So for example, between sodium cacalate of 300 and sodium cacalate of 250, you see the correlations what? They are literally identical to each other. You can see you can go all the way down to sodium cacalate of 50 millimolar, and it's basically the same. It's R squared of 0.96. So already I knew this was not true. It's not true that you need sodium cacalate of 300 for this experiment to work. It works identically at lower concentrations. If you go back and actually look at that original paper and look at the figure itself does not support that conclusion. So I'm not really sure why they did this, but anyone who is worried about this, at least for in vitro experiments, it's not true that you need 300. So here are some other things just to make sure we weren't crazy. So I tried KCl, which is of course not a buffer, and you can see it looks like garbage. It is extremely low. And then you can see for HEPEs 50, it starts to look a little bit worse. And for tris 50, which is a weaker buffer at this desired buffer pH of 7.4, it also does not work as well. So I was like, well, okay, well, previously I've done these experiments at 100 millimolar buffer back when I was a postdoc. And then, I was looking through some research, there, of course, people have looked at this like 20 years ago. And yeah, it turns out monovalent buffer concentration is also extremely important. So you can basically saturate some tertiary contacts without magnesium and they will still form. So just FYI, for anyone who's looking at and doing these experiments, the buffer is actually extremely important. So I found that as you change the buffer and decrease the buffer concentration, the change in mutational fraction of the tetraloop receptor goes up in magnitude. So at 300, they're basically identical. 250, 200. At 50, you can see a large difference between the 40 millimolar magnesium and the 0 millimolar magnesium. And what does that profile look like? It looks like this. So this is at 50 millimolar sodium cacalate. You can now see the expected change from going to basically no reactivity to high reactivity. So kind of the mini conclusion here, and this has been shown previously in literature, I was just unfortunately not super aware of it, that you can saturate basically most tertiary contacts, other than like specific metal binding ones, like the metal core receptor, with sodium cacalate, and thus with 300 millimolar sodium cacalate. And thus, anyone who's doing these experiments and is looking at tertiary contacts, you are totally overstabilizing your tertiary contact and probably other features too. Okay, and it's completely unnecessary to do. Okay, sorry for that tangent, but that was way too much time for me to figure that out. So now everyone else has to experience that too. Okay, so so just to make sure that I'm measuring the right thing, I created 2 knockouts. I created a tetraloop knockout where I mutated the GAAA tetraloop to a UUCG tetraloop, and then a TLR tetraloop receptor knockout, where I mutated the tetraloop receptor to just a helix. So now we can be sure that the change I'm observing is actually, oh, sorry, I forgot to update these. So here is the wild type at 15 millimolar concentration without magnesium. You get a reactivity of approximately 0.05 for the receptor, approximately, a range of 0.2 to 0.3 for the tetraloop. In the UUCG mutation, it has no ability to form it only has the receptor. The receptor has the identical reactivity to the no magnesium. In the no tetraloop receptor, the tetraloop has the same reactivity to the no magnesium. Again, this is clarifying and making clear that I am observing the complete formed and the complete unformed conditions. So again, based on these mutations. So again, this is super important. Okay, now that I beat that dead horse, let's go forward. Okay, so let's just talk a little bit more about the tetraloop receptor. This tetraloop is a GAAA, shown in red here, and then it has 3 A's of interest in the receptor. There's A4 and A5, which are forming what's called A platform. It's a very interesting, unique structure where basically the 2 A's come together and are like doing this. And then there's A8, which is sitting on top of it, and that's forming a direct hydrogen bond to one of the A's in the tetraloop receptor. So for each one of these, I wanted to see how are these nucleotides behaving as a function of magnesium. So here we're looking at the 3 A's. I'm averaging together because they basically have the exact same behavior. So you can see in the wild type, in the presence of the receptor, there is a rapid decrease in reactivity, going to basically 0. Where in the case where there is no tetraloop receptor, you see there is basically no systematic change. It's basically a straight line. So again, this is what we would expect. Completely consistent that without the tetraloop receptor, it stays about the same reactivity. When you have the tetraloop receptor, it docks in and thus becomes protected from DMS. What about on the other side? So for A4 over here, you see, again, this rapid decrease in reactivity as we saw for the A's. But what we see for the knockout, so without the receptor, without the tetraloop, we actually see an increase in reactivity, which really surprised me. I wasn't really sure what was going on here. It doesn't go up that much, but it's interesting. Similar for A5, we see something very different. So for A5, we see a kind of decrease initially in the presence of the tetraloop, but not nearly as much. And it kind of has this weird pattern that I don't fully understand. Maybe it's just error. And then on the without the tetraloop, it's it's similar. It doesn't have this very quick decrease, but it's basically a plateau and the reactivity doesn't change very much. On A8, so this is the one forming direct interactions with the tetraloop. You can see again this rapid decrease. Again, makes sense, going to basically 0. But then without the tetraloop, you see a rapid increase. Now this really confused me. Like, why would you see a rapid increase? That doesn't make any sense. So I thought a lot about this. It's like, does this data make sense in the context of what we previously know about the tetraloop receptor? Well, there is one solved structure of the tetraloop receptor without the tetraloop. So, and also happens to not have magnesium either. And this was a structure solved by Julie Fagan by NMR long time ago in the 90s. So again, this structure I'm showing you on the left has no magnesium and no tetraloop receptor. Sorry, no tetraloop, sorry, no GAA tetraloop. And if you see the structure, it looks really odd. A4, A5, A8, they're all kind of stacked into each other. You know, when I first saw this, I was like, can this really be real? I don't know. I don't know if it's real. It's an old structure. But it was interesting to me. Some of it makes sense because it doesn't have the A platform. A platforms appear to only form in the presence of magnesium. This has been known for quite a bit of time. So it's at least plausible. So what I was thinking is maybe what we're observing at the beginning is actually a structural change based on magnesium. Maybe when you add magnesium, you allow the A platform to form. And that's the change we're observing without the tetraloop. Then you have a secondary transformation. Of course, you have now the conformation with magnesium. And then now you have the tetraloop dock in. So what I'm suggesting is maybe it's forming a structure not exactly like this. Obviously, everything's dynamic, but I'm just simplifying it down. It allows for the A platform to form. And then the tetraloop can now nicely dock into the structure. Okay, well, how can we look at this? Very simplified model. Well, so again, it's a 3-state idea. Basically, no magnesium. All the A's are inside, have similar chemical environments. Then with magnesium, we have the A platform form, and now A8 can sit on top of it. And then we kind of have the tetraloop dock in. Well, we can look at the changes in reactivity in each case. We can measure each of these conditions, right? So no magnesium, no GAAA. That's just the first point without the in one of our mutations. And then we can see the case with magnesium without the tetraloop receptor. Again, that's just a case where we take one of the mutations. And then the form complex, where you have magnesium and the tetraloop. And you can see that each one has its own kind of profile. Now, again, this is very simplified. There's only 3 points. So what I was thinking is, well, we have 3 models to explain this. Can we just look at the solvent accessibility for all 3 states? And you get something that looks like this. So what really struck me in this is what's going on with A8. So A8, it starts rather low in solvent accessibility. If you remember, it's just in the helix. Then when you allow the A platform to form, it has very high solvent accessibility because it's sitting on top of the A platform. Then it drops back down when you bind the tetraloop, because now the tetraloop is hiding. Sorry, these are of N1, the N1 position. Sorry about that. This is where the chemical modification occurs, where methylation occurs. Okay, you see a similar thing going on with A5. A5 generally stays the same throughout. You see basically the same effect. The only one that looks kind of different is A4, where A4 drops to 0. We see that, but we don't see this change in reactivity from the in addition to magnesium. Again, I'm not claiming this is perfect, but I think it gives you some insight to what you can measure with DMS reactivity compared to other features like solvent accessibility. Clearly, solvent accessibility is not it, and we only compared just one state for each one. And let's not forget that RNA is extremely dynamic, and so there's likely ensemble states. Okay. What about actually measuring the stabilities? So the first simple test I did was I took the original wild type structure and I inserted 3 base pairs into all three helices. So this is likely to cause a destabilizing effect because basically with each of these helices, we are now turning the tetraloop away from the receptor. So thus, if this measurement system works, we would expect to see a decrease in stability in each case. So this is what we see. So the wild type, so note we are kind of changing the axis a little. It's easier to plot it this way and to do the fitting. So the fraction GAA DMS protection. So basically 1 protection means it has no reactivity, and 0 protection means it has its original reactivity. So what we can now measure is what's called the mag1/2. Mag1/2 is the concentration where half of the tertiary contacts of all our molecules are formed. So the wild type has a mag1/2 of 0.22. And in this mutation, when you insert this 3 base pairs, you see it now goes up about 5-fold to 1.1 millimolar. So a rather significant destabilization. You can also notice, this is the other really interesting thing, that you can no longer reach the maximum protection. So that means that, you can see in this orange curve, it only goes up to about 0.7. So that means there's still reactivity compared to the wild type. So to me, this suggests that the tertiary contact, it's just not formed 100% of the time. It's formed like, I don't know, 70-ish. I don't know what the fraction is. So I thought this was extremely interesting. You can do the 2 other insertions. You can see, for example, when you insert into helix 2, the effect is even larger. It's now 1.37, where if you insert into the 3rd helix, it's actually not that bad. It's only it only is about a 3-fold effect. And this makes some sense, where you insert, of course, is going to play a very big role. Also, previous studies have shown that this motif right here has the ability to concat, and so it can flip it out its nucleotide. So maybe it can just compensate for this mutation. But I think the takeaway point here is that this technique is rather sensitive and has the ability to make really nice measurements for tertiary contact stability. Okay, so what about more? So one of the critical things to do is let's compare this method, which I'm calling qDMS-MaPseq, quantitative DMS-MaPseq, to existing mutations. So that paper that I showed at the very beginning, that had Dan Hirschlag and Will Greenleaf, they ended up measuring about 1,500 different mutations. They found that there were some mutations in the tetraloop receptor that behaved very similarly to the wild type. So those are the ones I selected from. So I picked about 100 different ones. And so for each one, I'm just inserting them into my scaffold and then doing this magnesium titration to see what their mag1/2 are. So here's just an example. There are ones like mutant 1 here that behave basically identically to the wild type. And then we have ones such as, again, 100, this one down here, where they have significantly higher mag1/2s, indicating they're much less stable. Okay. But how do these mag1/2s compare to these delta G measurements made by the platforms called the RNA-MAP platform by Will Greenleaf and Dan Hirschlag? Well, it correlates pretty well. So I had to take the ln because likely this mag1/2 is some kind of rate constant or equilibrium constant. So if you take the ln of it and compare it to the delta G, the known delta Gs, you can see I can get a R squared of 0.64, which is pretty good. Strong correlation. Again, this experiment can be done really easily. You can do it in about 5 hours, and it's very accessible. Also, keep in mind the buffer conditions were different. There's lots of different things about these 2 experiments. But what was really surprising to me, what I think is extremely interesting, is you can just compare the reactivity at a specific magnesium concentration. So here I'm just showing magnesium. So here is the ln of each of the average of the 3 A's together, their reactivity values of the GAAA at magnesium of 7.5. That correlates even better to the delta G. And A, that's only one experiment. You don't have to do all 16 points. But B, this really says something about what reactivity actually is, right? So we treat reactivity as a pretty nebulous value, as it's either low, it's in a base pair, or it's high, it's not in a base pair, right? And that, that works great. But I think this actually shows that the reactivity has specific meaning. Like in this case, I think it is directly correlated with whether the tertiary contact is forming or not, meaning it is somehow reporting directly on either the solvent accessibility or some other related faction, and that the individual values have meaning, not just high versus low. Now, you might be like, oh, you just cherry picked the best one. No, you can see at different magnesium concentrations, it still is correlated. Obviously, it gets worse and better given like there's a sweet spot, but it's not that I just picked one value and it was the one value that worked out. Many different values seem to work. Obviously, if you go down to very low, it doesn't work as well. But so this value might be different for different tertiary compounds. Okay, I just one quick last thing to show you. The last thing I want to talk about is what other types of information can you extract from these from qDMS-MaPseq, from these DMS values. Well, I want to talk about two cases where you see very interesting mutational profiles, reactivity profiles over different magnesiums. So I'm going to talk about two different ones that I find very interesting. The first one is a CC mismatch mutation. So basically where these arrows are, there's usually a GU there. But a very common mutation from the pool I was looking at was to replace this with a CC. So I already find that crazy that you can just swap out a GU with a CC mismatch and it still is fine. The reactivity, the delta G and mag1/2 are very equivalent to the wild type. But the thing that really was surprising to me is if you look at the reactivity, again, red is high, blue is low. The reactivities of these two Cs without magnesium, i.e. the tertiary contacts not formed, are relatively similar to each other. But at 40 millimolar magnesium, they're completely different. The reactivity on this C is extremely high, higher than it was initially, and its C partner is basically 0. It's you would think it is a Watson-Crick pair. So what's going on? Well, I did some 3D modeling using FARFAR to build a model of this, assuming that the orientation is the same as the wild type. And again, the reason I believe this is a fair assumption is because the binding mode is similar based on this previous paper by Dan Hirschlag. And you get something that looks like this. So coloring is the same. This is the CC pair over here. These are the A platform's a little messed up. And there's a really striking feature you see in the CC. So I'm going to zoom on in. You get this, and this is a pretty common feature in the top 100 models I looked at. The top 10 models I looked at, is you see that there's this asymmetry. One C had this, so right here, this is the N3 position, these two positions. That's where DMS reports on. So on this C, there is a hydrogen bond, very prevalent hydrogen bond here. And this other C is now locked in this turned position. This would very nicely explain this asymmetry, where for this C, there's going to be no reactivity, and for this C, there's going to be very high reactivity. So I actually have seen a similar case. That's what so excited me about this model. There is another CC pair I've seen before, has basically the same interaction. But this comes from a crystal structure. This comes from the kink turn from the mini TTR construct that I'm using. And so this CC, it comes from a crystal structure. And you see it also has this asymmetry, and it also has the same reactivity pattern, where initially the two Cs are more comparable, and then you get this sharp change with magnesium, where you see a totally, like, the reactivity of this one becomes many order, like 15 times more than this one. Okay, one last one I want to show you, I know I'm running a little bit over time, is there's this other construct that has a inserted A. So basically, instead of these these three A's, there's now a fourth A that comes right before A4. So if you remember these three reactivity profiles I showed you before, where you have A4 and A8 doing this crazy drop in reactivity, and then you have A5 doing its own thing, basically not really changing too much. Well, this new A looks completely different than the other A's. So it does undergo a sort of rapid decrease, but then it platforms out and never goes down any further. So this again, really, as you can see, and so you can see it's pretty similar in each case. So what's going on? Well, if you look at the models I generated, the one to me now, again, this wasn't the lowest energy model. The thing that was the one that makes most sense to me is for the A to be completely flipped out. This allows the other A's to perform in the exact same way that they did initially, and this A to have this very interesting reactivity pattern is that it just levels out, basically, like a flipped out nucleotide. And this trend looks very similar to other flipped out nucleotides. Okay, that's it. So I hope people took away from this that first, buffer conditions are extremely important. For tertiary context, probably for most things, but for tertiary context, make sure you have the correct buffer. That DMS chemical mapping with magnesium titrations is an extremely powerful and easy way to measure the thermodynamics. These measures are high quality and agree with previous delta Gs, and the DMS can give you nucleotide resolution, which you could not get from these previous methods. And just like this, you could just like before, you can super high throughput them. I have libraries that I'm making now that are 100,000 different mutations, and you can do the same exact experiments. All right. With that, I'd like to thank my group. Here is us going out to dinner over the summer. And I would like to thank both NIH and NSF for the grants that help support this work. And I would be happy to take any questions. Thank you.
Thank you very much. Joe, do we have any questions in the audience?
I can get us started with one question. I'm curious, kind of in the spirit of like blind prediction challenges, how do you see these sort of stability measurements playing into that? How could you think about kind of, would this help in predictions? What are your thoughts?
Yeah, absolutely. I mean, definitely with, the rise of all these, with deep learning, of course, that's something I'm very interested in. And because it's so easy to multiplex these, I mean, I'm hoping, again, to try basically every mutation, every single tertiary contact that we know of, and every possible mutation, which hopefully we can connect with deep learning to help make better and better predictions. But yeah, I don't know. I don't know what will be the magic bullet. I mean, a lot of it also has to come to do with the flexibility of the RNA. Can the tertiary contact actually reach each other? And like that's a very hard problem that I'm also working on too.
So this is kind of what I wanted to ask. Do you expect some of the contacts that you will not be able to see with this method? Would be that some kind of characteristic that would prevent?
Yeah, so I mean, there are definitely, so there are tertiary contacts that are too weak for me to see. You just basically see no change. There are also ones where you've changed the topology significantly enough where the two ends can't reach each other. And then no matter what you change the condition, it will never form, basically. So yeah, there's of course, I mean, all experimental procedures have limits, and there's definitely the range. I mean, I didn't talk about it, but the range appears to be a little bit lower than that previous experiment, about 1 kcal per mole less. But that's fine. It's still a really nice, like 5 kcal per mole range.
Questions from the audience? Not yet. So then I can ask you about the Ribonanza. Did you participate?
I did not. I was thinking about it, but I just ran out of time and didn't do it. Yeah, I think it's a very interesting idea. I think we should be very careful about our conclusions from this initial test, and we should worry a lot about data quality and what type of RNAs we're looking at. So I would, you know, before this thing was announced, I have put together a set of about 500,000 RNAs, and with chemical mapping profiles. And you know what? Based on which RNAs you pick, you can make any technique look good. So it's very, I think, very important. Without going into super much detail, it's very important to pick a good set and be very confident in the reactivity values associated. Oh, there's some
yeah, there are some questions. So I would just ask, do you do the experiments like in the lab, or you outsource the experiments, like as you said?
I did in the lab. Yeah, they're very easy to do. Yep.
Okay. We have one question from Asta. Hello, Asta. How far tertiary contact can be predicted? I'm not sure in which. What far in sequence?
Far.
Yeah.
Yeah, I guess I don't know if you want to follow up with. I don't know what far means. If it's a distance thing, then of course it's really dependent on like the internal flexibility of the RNA. I mean, if the RNA is very flexible, then it'll be able to form much further. If it's not very if it's very rigid, it'll be much harder.
Okay, there is a question from Sikiera. Nice talk. Any plans to use shape map to cover all 4 residue? And my follow-up to that, can you a little bit mention about this TMS for the rest 2 residues? Because I think this is related. I think this is what you mentioned.
Yeah, so so definitely very good question. I don't really like shape personally that much. I will be the person to say that. I think shape is fine, but I have a much harder time interpreting what the reactivity values mean. Again, if you're just looking at a change, then it's great, right? So if you're just wanting, you know, you have the tertiary contact formed and unformed, then yeah, it doesn't really matter. You can use any chemical mapping technique. I personally find shape map to be harder to do. I've had less success with it than DMS. I am more interested in, as Magnus said, the 4 nucleotide DMS chemical mapping. Again, this was developed by Weeks and Musto. And basically they showed that if you change the pH slightly, you can about 1% or 0.1% modify G's and U's based on deproteination. So basically G and U becomes deprotonated, and thus there's a chance for it to become methylated. Seems to work really well. I mean, Ribonanza, I used it. Again, you need more reads, but it seems it seems good. I've started to use it myself. It seems fine. I prefer that over shape at the moment. But again, if you can do shape, there's nothing wrong with shape. It's just a personal preference. Yeah.
There's one more for Megan. Very cool. Is it possible to get kinetic information about tertiary context formation using this method, or those timescales too small to capture?
Yeah, great question, Megan. Yeah, I don't think so. I think these are all thermodynamic, right? So I think the reactivity, it's just too slow. DMS is too slow. I know Kevin Weeks developed a new shorter time scale one that may be possible. This is something I'm super interested in, but yeah, I'm not really sure I'm set up to do it. If you're interested in such techniques, you should check out, oh man, I'm blanking his name. He was a postdoc with Julius Lux, and now he is at Buffalo. I don't know why I can't think of his name. That's really sad. He's a super awesome guy. But he's doing basically he's doing 1 nucleotide at a time chemical mapping. So he can stop at every nucleotide, and then you can sort of figure out some kinetics, although it's not perfect. Yeah, sorry. I feel really bad for not remembering his name.
We can send it also later if you want. So yeah. So maybe one, and so this is also what I wanted to ask. So it is related to molecular dynamics, which was like your background for your PhD. So when from Valid, Wonderful talk. When swapping nucleotides to measure and compare delta Gs, did you look into molecular dynamics simulations to confirm or not confirm results, such as pool or umbrella sampling simulations?
Okay. So no, I did not do that. I guess I have a slight issue with your wording of confirm. I don't know if molecular dynamics confirms anything. I think it is a great way of trying to understand mechanisms. But as someone who has spent a lot of time in molecular dynamics, I'm not really sure it's good for that. Now, it's great for trying to build up like what's going on. Like, is the tertiary contact just falling apart? Is it like one nucleotide at a time type thing? That can allow you to come up with new experiments to test. But yeah, I'm not really doing that anymore. It's definitely, yeah, I definitely have some interest in it. But at this point, I would probably collaborate with someone who knows more about it than me now. It's an interesting idea, but again, I think you have to be very careful with what you're trying to what are you trying to do. And by the way, Megan, sorry, it's Eric Strobel. Eric Strobel is the guy's name. Very cool guy. Definitely check out his work. So yeah, hopefully that answers the molecular dynamics question.
Okay, do we have the last questions? Yes, some thank yous in the chat. If not, then Joe, good luck with your lab. Hopefully we'll see each other soon at some point. We are relatively close to each other now, and thank you for the talk. Talk is recorded, will be on our Slack or our YouTube, and we see each other, I think, in 2 weeks.
Yep, sounds good.
Thank you very much.
Take care. Thank you so much.
Have a good week.